let's start with functional plasticity. And we cannot avoid something that is very fundamental for every neurobiologist, everybody who's interested in plasticity and learning in the brain, is the Hebb hypothesis. So Donald Hebb, Canadian, a psychologist, wrote in 1949, a very influential book, and there he said the following. That's very important to understand what he said. He said about plasticity and learning in the brain, it was his hypothesis. He didn't know whether it exists or not. He said, when an axon of cell A is near enough to excite cell B, or repeatedly and consistently takes part in firing it, so cell A consistently is involved with activating cell B, not on his own, but together with others, consistently, again and again, he says, some growth or metabolic changes takes place in one or both cells, A and B, such that A's efficiency as one of the cells firing B is increased. Or we like to call it today, or to summarize the Hebb hypothesis today, fire together, wire together. What does it mean? Hebb says the following in simple words, that if cell A is involved with firing cell B, again and again, so I'm cell A, I'm active, cell B is active following me. Not only me, but with other friends. But again, I'm active, cell B is active. Active means firing a spike. I'm firing spike, cell B firing spike again and again. Sufficiently enough time, some changes between these cells takes place, and eventually cell A becomes more effective in activating cell B. It's an hypothesis. We don't know that it's true or not, but it was a very formative hypothesis. Very, very important hypothesis. Something about the causality that I am, as cell A, succeed causally to cause you to fire cell B again and again, then we are becoming good friends and we strengthen the synapse among us. So in the last 20 years, a lot of experiments have been done, and indeed we found out that Hebb's rule, or Hebb's hypothesis, is indeed implemented in some, both hippocampus, and also cortical cells, showing that synapses, which connect cell A and cell B, are very, very, very plastic. So let me show you what we succeeded to show. Of course, when I say we, is the community of neuroscientists. So again, just to remind you, we know that the axon of cell A fires an action potential, a spike, all or none. Remember the Hodgkin-Huxley model. We understand the spike very well. Cell B, the postsynaptic cell, responds with an EPSP, excitatory postsynaptic potential. Let's say that this is before learning. That's the basic efficacy of this synapse. This spike, 100 millivolt spike, generate this little EPSP, let's say 2 millivolts EPSP. After learning, somehow, we'll discuss how, somehow Hebb says that when cell A fires cell B sufficiently time, something about the connection is changed, and cell B, and the connection becomes more effective. So after learning, Hebb says, after learning, this spike increases the probability of cell B to fire. How? Something becomes stronger, the synapse becomes stronger. For the same spike, you get a bigger EPSP, a stronger synapse, more depolarization due to the same spike. So this is actually, in terms of synapses, spikes, and EPSPs, what Hebb is saying. If this is active enough time, then the weak synapse becomes a stronger synapse. I want to show you what I just said schematically in the following way. What could be a mechanism for such a thing? What could be the mechanism for making the synapse stronger? We know today that one possible mechanism, is that the synapse becomes more effective for a given spike, making more potential postsynaptically due to the insertion of new receptors in the postsynaptic membrane. So you remember that there was a spike here, there is a release of transmitter, and there is a receptor there binding the transmitter, opening channels, making an EPSP here. Okay? So the spikes makes an EPSP. We know today that due to plasticity of the synapse, of this amazing plastic device, the same synapse may have now postsynaptically more receptors. And I'll show you what is the mechanism for it. More receptors postsynaptically. So again, the same spike, the same presynaptic signal generates postsynaptically a larger EPSP. So I would call this a weaker synapse, and I will call this a stronger synapse. And the mechanism for making the synapse stronger is that for the same spike, the same transmitter release opens more ion channels postsynaptically, thus generating a larger EPSP. Okay? So this is a possible mechanism. I could give you another possible mechanism that presynaptically, the same spike would release more transmitter. Then, for the same spike, I would get more transmitter release, and this then will make the synapse also more effective. So this is a postsynaptic mechanism to make the synapse more effective. How do we study what really happens in synapses? And this is the idea. The idea is that today, with new techniques, we can record both in the in vivo case, in the whole brain, but typically in slices. You can record from two cells in a brain slice, for example, in the cortex. You implant an electrode here and here, in these two cells, and you start to ask the question, what makes this particular synapse, if they are connected synaptically between here and here, what makes the synapse between these two cells more or less effective? What could make the synapse more or less? So you really can stimulate the blue cell, you can record synaptic potentials in the red cells, and you can try to manipulate things until you find what could be the mechanism for synaptic plasticity. What are the conditions? What are the conditions for the synapse to become stronger, to have more receptors, to respond stronger to a spike? What could be the conditions? Now you can manipulate each cell separately, and so you can control the condition and find out what are the conditions. So this is what experimentalists do today, did about 12, 15 years ago, and they found something very, very important, very, very fundamental, a mechanism that is called spike timing dependent synaptic plasticity. Spike timing dependent plasticity, or in brief, STDP. They found that this particular condition, that I will just show you in a second, this particular condition makes the synapse, the connection, stronger. So here is the condition. Here is your synapse between the cell A and the cell B. And you want to make this synapse stronger. You want to generate a larger EPSP at the postsynaptic side due to the spike in the presynaptic side. And that's what you do, apparently, in order to make it stronger. You activate first the presynaptic cell. You generate a spike there. You with your electrode. Spike here, and then with the other electrode in the postsynaptic cell, you generate a spike there. So there was a spike here and a spike there. And again, a spike here that you generated, and a spike there that you generated. So presynaptic spike, postsynaptic spike. Presynaptic spike, postsynaptic spike. Enough times, again and again, and suddenly, after this enough repetition, the synapse becomes stronger. You get a larger EPSP, as I showed you before. This is called spike timing dependent plasticity, because it depends on the timing between the presynaptic spike and the postsynaptic spike, presynaptic spike, postsynaptic spike. In this order, pre, before, post, you get enhancement, what we call long-term potentiation. The synapse is potentiated, becomes stronger, potentiated, long-term potentiation, LTP, for a long time. Long, it could last minutes, hours, maybe days, maybe lifetime, but long. So after this kind of pre-post enough time, you get an enhancement of the synapse. Let me show you real experimental results. So in this case, I should have mentioned here that if you do it in the reverse order of timing, so you first activate the postsynaptic cell and then the presynaptic cell, so post-pre, then the synapse becomes weaker. So that's anti-Hebb, because Hebb said only in one direction, that when A is active enough to activate B, then the synapse becomes stronger. But now I show you that the synapse may also become weaker if the reverse order in spike timing takes place. So here is the experimental result, one of the experimental results, in the cortex in this case. So here is the case where the postsynaptic cell fires before the presynaptic cell. The postsynaptic cell fires presynaptic. And this, I said, this order makes the synapse weaker. So this is our control strength. And you can see that for this order of timing, the synapse is less strong. The EPSP is less than the normal. And in the reverse order of time, you see here, this is pre, before, post, pre, before, post, again and again, the synapse becomes stronger than control, maybe twice than the control. Okay? So this is the LTD, long-term depression of the synapse becomes weaker. And this is the LTP, long-term potentiation. The synapse becomes longer, more stronger. Look at the time window for this mechanism. It's a short time window, whereby if the presynaptic spike starts before the postsynaptic spike, you get LTP, long-term potentiation, about 40 milliseconds time window. If the difference in timing is longer, so pre, and then you wait a minute, and then post, the synapse doesn't care, it doesn't change. And also the reverse. If the postsynaptic cell fire before the presynaptic cell in this time window, the synapse becomes weaker. So it's a very narrow time window for these mechanisms to work. It doesn't explain many other things. For example, the Pavlov case, where the dog hears a bell and then sees a food. The difference between bell, food, until the bell itself generated the saliva could be seconds. So this mechanism is millisecond mechanism, much briefer. And what we do, what we know about learning, for example, this classical Pavlovian learning is much longer time scale. This doesn't explain Pavlovian learning. We still have to understand long-term mechanism for plasticity. How do I learn something from childhood and remember it when I'm adult? This does not explain everything, but it is a very important mechanism for rapid learning. Now, the synapse becomes weaker or stronger on this time scale. It's a very important time window. And today, because we have these functions experimentally, we can write the equations that describe this LTP, long-term potentiation, when A fires before B, or long-term depression when B, the postsynaptic cell, fires before A. We can write down equation, mathematical equation, that explains, that describes this function. We have mathematics, a model for plasticity of the synapse. It's very important now, because now when I have a model, I can really do theory or even generate machines that follow this rule, the machine. And maybe the machine will start to learn. And we shall discuss at the end that today what is called in computer science, machine learning, the whole aspect of machine learning is inspired in many ways by the brain, because the brain is a machine that learns. This is one mechanism, the spike timing dependent plasticity, this causality relationship between pre and post or vice versa. And since I can write an equation, I can develop machines that follow this equation, like my machine follows this equation, and then we can apparently learn. Just to summarize this part about spike timing dependent plasticity, of course, one wants to really pin down and understand what is the mechanism in the synapse that is sensitive to the timing of the spike pre and post. Why, when there is a spike in the presynaptic membrane, and then there is a spike in the follower cell, in the postsynaptic cell, what happens really physically? What is the mechanism that enables this very sophisticated machinery to become stronger or more efficient? There is a lot of focus today on this very fundamental issue about the brain, about synaptic plasticity. We know a lot about mechanisms in the presynaptic part, not only the vesicles, but a lot of other mechanisms are embedded here. And also in the postsynaptic part, not only the receptors plays a role, but all this recycling of receptors and all these other things that are really beyond this lecture. But you should know, of course, because synapses are so important, probably the most amazing plastic mechanism in the world. They learn, they change very fast. They have rules for learning. And this learning certainly underlies your capability now to learn something new. So we must understand what makes this fantastic synapse so dramatically capable to change, but also clinically. We know of diseases, like Alzheimer's, whereby the synapses stopped functioning correctly at early stages of the disease. And so you start not to be able to learn and remember. And so we must understand the mechanisms at the level of synapse and the level of spikes and the level of postsynaptic potentials in order to understand these amazing plastic mechanisms of synapses. So this was functional plasticity, and now I want to go to structural plasticity.